I'm Hardeep from the Australian National University. Today I will discuss our recent work describing microchromosomes as building blocks of bird, reptile, and mammal chromosomes. I've changed the title of my talk to correctly reflect the results of our work. I want to begin by acknowledging the traditional custodians of the Canberra region where I live and work and pay my respects to their elders past and present. I want to thank my collaborators on this project, especially Paul, Aurora, Arthur, and Jenny. Preprint of our work has just come out and it can be found at bioRxiv for details. This work relied on the availability of high quality genome assemblies provided by the Australian Amphibian and Reptilian Genomics Consortium, DNA Zoo, and other individual genome projects. We would also like to thank National Computational Infrastructure in Australia, which provided 2 million CPU hours required for performing this analysis. And last but not least, we as a team thank our funders for their continued support. So, microchromosomes are small, tiny chromosomes that are barely visible under the microscope. They have gained renewed importance, and many investigations are trying to understand their properties in recent times. One of the distinguishing features of microchromosomes is their abrupt size difference compared to big macrochromosomes. Karyotypes of representative animals, including chicken, alligator, python, and turtle, clearly show that microchromosomes are a feature of bird and reptile genomes. Macros are shown over the pink background and micros in light blue background in these karyotypes. Lizards are slightly odd in that they do not have this clear demarcation of small and big chromosomes. This is, however, not true for all lizards. Some lizards do have distinctively small chromosomes. At this gross karyotype level, it is evident that microchromosomes are ubiquitously present in birds and reptiles. So we wanted to explore the origins of these microchromosomes. Are microchromosomes fragments of big macros as byproducts of degradation? Alternatively, were micros the ancestral state, giving rise to big macrochromosomes through fusions? There is also a possibility that micros and macros have coexisted in animals. In addition, we wanted to know if there are any mechanistic differences between micro and macro in bird and reptile lineages. To address these questions, we performed whole genome sequence comparisons of species listed in the phylogenetic tree on the left. We included 7 birds, an alligator, 2 turtles, 3 snakes, and 4 lizards to represent deep evolutionary history of amniotes. We also compared this amniote species with mammals, including platypus, koala, and of course humans, and Amphioxus to represent the distantly related pre-vertebrate state. We use the popular LAST-Z software, which is used by Ensemble and UCSC for comparative genomics. It is very good at detecting remote homologies between pairs of species. As an example, I show alignments between chromosome 1 of the chicken on the x-axis compared with the chromosome 1 of the green sea turtle on the y-axis. The left panel shows the location of alignments between the two chromosomes and heat colors representing alignment scores. There were about 160,000 alignments between the two chromosomes of varying scores. But we can see diagonal lines amongst all that noise, which is likely to be the true alignment between these chromosomes. These alignments are chained into longer alignments where possible, as shown in the middle panel. This chaining process reduces alignment numbers from 160,000 to about 85,000. There is still a lot of noise, but at least one diagonal line is visible. Subsequently, these alignment chains are grouped into syntenic blocks or nets, as called by the UCSC, which allows for smaller rearrangements and differences to be incorporated into a larger syntenic block. We can see that netting process reduces the number of alignment blocks to only 298 between these two chromosomes. We have used these syntenic blocks to infer evolutionary patterns between species in subsequent slides. I'll show several of these images, so let me walk you through some basics about what is displayed here. A horizontal line of segments represent the genome of each species. Macros are in pink and micros are in blue. All genomes are scaled to a uniform size for these images. Syntenic blocks between a pair of species are shown using ribbons. If the ribbon is between macros, they are marked in pink. Blue ribbons indicate microchromosomes, and occasional green ribbon indicates syntenic alignments between a micro of one species to a macro of another. Sequence comparisons of five birds towards the bottom of the figure shows one-to-one correspondence for micros and macros with only a handful of rearrangements. Two turtles at the top of the image also display nearly perfect one-to-one correspondence except for two green sea turtle micros that have parts aligning to macros in slider turtle. Comparisons between slider turtle and the emu genome show that all emu macros align to macros in slider with some intra and interchromosomal rearrangements. However, pattern is slightly different for microchromosomes between the slider and emu. We observe four emu micros that are fused into two macros in turtles and two other micros fused to terminal regions of macros in turtles. Based on these alignments, we conclude that macro and microchromosomes between and within bird and turtle lineages are largely conserved. Next, we look at snakes and lizards as a squamate group. Snake genomes shown at the bottom of the figure show that macros and micros are almost wholly conserved between three snakes. There were relatively few macro fusions or fission events and rearrangements between three species. The water dragon in the middle, which is more closely related to snakes than other lizards, has similar macro and micro to that of snakes. Apart from two micros aligning to terminal regions of snake macrochromosomes, there is one-to-one conservation between snakes and water dragon as well. Similarly, chromosomes of water dragon and teague are also one-to-one conserved. Two micros of teague seems to be fused into a macro compared to water dragon. Common wall lizard and common lizard, their genomes appear to be grossly conserved between the two, with few fission or fusion events between them. Excluding common wall lizard and common lizard, we can conclude that chromosomes are grossly conserved in one-to-one manner across squamate groups, and microchromosomes appear to be the ancestral state for chromosomes that appear as macro in common and wall lizard. Next, we chose emu, green sea turtle, Burmese python, and teague as representatives of their respective clades for comparisons at a deeper evolutionary timescales. To reiterate the results, all turtle micros align to a subset of 15 emu micros, marked with a blue line under the emu genome. Comparisons between python and teague have shown one-to-one conservation as well, except for two micros in teague fused to form a macro in python. Novel comparison in this figure is between emu and python in the middle. Syntenic blocks between emu and python are relatively smaller. However, we can still confidently deduce the pattern of micro and macro conservations between the two. Just like turtles, all python micros align to a subset of emu micros marked in black bar above the emu genome. It is evident that snakes and turtles have retained different subset of emu micros. The simplest explanation for this pattern is that birds represent the ancestral amniote state and snakes and turtles have lost some microchromosomes due to fusions independently. We extend the analysis to all versus all pairwise comparisons to confirm that the observations and inferences hold true for any pair of species. We calculated the percentage of microalignment lengths of query species that align to micros in a target species, which is shown here in the heat map. Query species are on the y-axis and target on the x-axis. As we can see, that heat map is largely blue for most cells, showing that microchromosomes of almost all species are aligning to microchromosomes of other species. The variability at a percentage level and asymmetry in the heat map is the reflection of selective retention of subset of microchromosomes between different lineages. Most important in this figure is the vertical blue zone of bird target species. 72 to 100% of microchromosome alignments for most species are to bird micros. So this figure shows that bird microchromosomes are likely to represent the ancestral state for all amniotes. Next, we wanted to explore the origins of amniote microchromosomes. There are examples of fish with microchromosomes as well, which suggests that perhaps micros might be the ancestral state to all vertebrates. Oleg, who is one of our collaborators, in 2020 performed comparisons between the chicken and the Amphioxus genome. I show the Oxford grid plot from their paper. Amphioxus linkage groups are shown on the x-axis and the chicken chromosomes are shown on the y-axis with micros at the top and macros towards the bottom of the image. Focus on the green vertical rectangle, we see that Amphioxus linkage group E has alignments with chicken chromosome 1, 12, and 24. Conversely, though, if we look at the brown horizontal box representing chicken microchromosome 17, we see that chicken 17 is homologous to Amphioxus linkage group H and no other regions of the Amphioxus genome. They showed that most micros of chicken were homologous to a single or at most two Amphioxus chromosomes. We used our whole genome alignments between emu and Amphioxus to see similar patterns as previously observed. The figure shows representative alignments of 4 emu micros to Amphioxus genome. We identified 9 micros that had one-to-one homology with the Amphioxus genome. There were 6 additional micros of emu that were aligning to at most 2 Amphioxus chromosomes, implying fusions in ancestral vertebrates. Similar patterns were observed for turtles, snakes, and lizards as well. Therefore, we conclude that microchromosomes in reptiles and birds are like the ancestral stage. Let's come back to exceptional species that we saw in the heat map. Eagle genomes have undergone remarkable number of changes where the basic micro and macro patterns we observed for other birds doesn't hold. As an example, the largest emu chromosome is broken into 7 pieces, giving rise to 2 macro-sized chromosomes and 5 micros, giving rise to a widely different karyotypic structure in eagles compared to other birds. Alligators deviate from one-to-one chromosome homology we observed previously between the emu and the turtle genome. However, alligator is not as crazy as the eagles though. We see the general pattern of macro of birds remaining as macro in alligators too, with some exceptions. To explore the fate of ancient amniote micros in mammals, we compared the genomes of emu with those of koala, human, and platypus. The sizes of synthetic blocks are relatively small between species. Also, we note that there were weaker and scattered conservation patterns between emu micros and platypus chromosome. It is hard to see in this figure, but 4 of the platypus chromosomes appear to be composed of regions with homology to several of emu micros. Of macros, perhaps in the reptile-like ancestor of mammals, micro-microfusions may have been common. Another observation we can make is that mammalian chromosomes are not as conserved as reptile and birds, even at a shallower time depth of about 170 million years. I hope I've convinced you that micro and macro chromosome structures of birds and reptile genome is highly conserved within and between lineages. Second, the bird chromosomes represent ancestral state for amniotes with selective retention of micros in snakes, lizards, and turtles. Third is that mammalian chromosomes are highly rearranged and that there is some evidence of microchromosome fusion leading to mammalian chromosomes. Most importantly, comparisons of birds with tiny chromosomes of Amphioxus by us and our collaborators show largely 1-to-1 or 1-to-2 homology, suggesting that small microchromosomes were the ancestral state. It will be a pity if I don't show the picture of chromosomes at a chromosome conference. So, to gain mechanistic understanding of it, if and how microchromosomes behave differently to macrochromosomes inside a cell, we looked at their locations in the Australian iconic central bearded dragon. In figure A of the image, we see mitotic chromosomes. Note how the microchromosomes are spatially segregated at the center of the image. Figure B shows the polar view of diakinesis during meiosis. We can again see the central location of smaller microchromosome bivalents surrounded by macrochromosome bivalents. Similarly, in figure C, the central location of microchromosomes stained in pink in interphase nuclei is prominent. So the obvious question is if they are physically interacting. Previous studies in emu and rattlesnake have shown greater amount of interchromosomal or transcontact probabilities for these two species. Figure on the left is data from emu contact maps where darker red colors at the bottom represent increased interchromosomal or transcontacts between microchromosomes. Similarly, in rattlesnake as well, increased interchromosomal contacts were observed for micros shown in darker blue colors at the top of the figure. We extend the analysis to include turtles, alligators, and teak, which all have microchromosomes, to see how well their microchromosomes interact with each other. For each species, their micros are shown towards the top right corner, and darker the red, greater the interchromosomal or transcontact maps. We see similar patterns as snakes and emu that we saw in previous slide, whereby micros show greater amount of interchromosomal contacts compared to macrochromosomes. Scatter plots on the left of the heat map also shows the GC content of microchromosomes to be generally higher than macros in all three species. Micros are shown in green and macros in red colored dots. Line plots right of the heat map shows distance in megabase on the x-axis and contact probability on the y-axis. As group, we observe that microchromosomes generally have higher distance dependent contact probabilities compared to macros in respective species. From this analysis, we can conclude that microchromosomes with higher interchromosomal contacts is a feature of all birds and reptile genomes. Next, we looked at Python. The alignment plot on the top shows that few of the emu micros have fused to terminal regions of macros in Python, represented with green ribbons. So we divided the Python genome into micro, macro, and new macrochromosome states. Then we compared the GC content of micros in green dots, macros in red dots, and new macros in purple dots. The GC content difference between the micro and the old macro is very clear. However, the purple dots representing new macros are distributed among red and green dots, showing the transitory change in GC content for new macros. Similarly, in high-C interaction plots, we see that interaction of macros with some micros is relatively higher, shown in green arrow. Also, the distance-based probabilities of high-C interactions for new macros is like macrochromosomes overlapping with green line in the plot on the right. In conclusion, micros after fusion with a macrochromosome in Python retain higher GC, higher trans contacts, and higher distant dependent interaction probabilities like typical microchromosomes. Eagle genome has undergone significant rearrangements compared to other birds. This provides us the opportunity to look at the fate and properties of micros in much more detail. When we look at the new current state of micros and their GC content, we do not see an abrupt difference in GC content between micro and macro chromosomes. However, when we break the genome down into ancestral state as observed in other birds, we can start to see that old larger chromosomes split to form new microchromosomes retain their ancestral GC content, and older micros fused to form macrochromosomes retain their higher GC content typical of microchromosomes. Overall, we found that GC content reflected the ancestral state even after fusion and fissions. In contrast, the distant dependent contact probabilities of four classes did not consistently reflect their ancestral state. Ancestral big chromosomes displayed lesser, and microchromosomes displayed greater distant dependent interaction probabilities. Micros that have been fused to form macros retain their higher distant dependent probabilities like the ancestral state. Surprisingly, though, the new micros derived recently from a big chromosome attains elevated distance dependent interaction probabilities. So, to conclude that part two, microchromosomes occupy central region in the nucleus of cells and display higher trans interactions amongst them. GC content and trans contacts generally retain over a longer period but erode eventually. New microchromosomes acquire higher distance-based contact probabilities. So to bring it all together, we propose the following model of chromosome evolution that we see in extant bird, reptile, and mammals. Ancestrally, all chromosomes were small and tiny, microchromosomes without too many repeats. Subsequently, some of microchromosomes fused with each other to form larger chromosomes, followed by expansion due to genome doubling and accumulation of repeats and transposable elements. These repeats provided substrate for further micro-macro fusions with a subset of chromosomes left alone due to the lack of repeats and shielding provided by stronger interchromosomal interactions. Big macros with higher abundance of repeats and lack of trans interactions paved way for further rearrangements between macros. Macros, of course, kept fusing with each other, leaving micros alone that we currently see in birds and reptiles. With that, I'll conclude my talk. It is late at night for me, so I won't be able to take live questions, but please feel free to send us questions through emails or GitHub. Thank you.